Hey guys, welcome to AP Biology once again, and we are going to be talking about carbon and the role of macromolecules in preparation for doing a lot of stuff over the weekend and into next week in preparation for our test on Thursday on chapters 2 through 5. All right, so first of all, organic chemistry is the chemistry of life, and it's basically the chemistry of carbon too. And as I say up here, organic molecules can be really small, such as methane, which we looked at the other day in class, and huge, like a DNA molecule, which can be almost 7 feet long, yet it's packed up very tightly inside the nucleus of every single one of your cells. But they all have lots of things in common, all organic molecules, and as we did learn the other day, that chinops, the carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur, and some other things are all going to have some combination of those. But in order to be organic, a molecule must have carbon and hydrogen. Okay, must have carbon and hydrogen to be considered organic. So, one of the interesting things that came about as we started learning about evolutionary biology and genetics and things of that nature was how did these first macromolecules come about? And there was a couple hypotheses out there that were actually put to the test in 1953 for the first time by Stanley Miller, where they basically built a model system that simulated the early Earth's atmosphere and oceans. And they were able to run this for several weeks, and they were able to synthesize some basic organic compounds. And so here's the setup right here. They basically had an ocean that they were heating up, and then with a heat source, and then that evaporated water would flow up into this atmosphere. In this atmosphere, they put some gases. They put some methane, that's the CH4. They put some ammonia, which is NH3, and they put some hydrogen, and they added a little spark of lightning. And again, this is a approximation of what the early Earth's atmosphere was like, and how do we know this? Well, we know there was no oxygen in the early Earth's atmosphere because there's no evidence of oxygen in the geology, i.e., there's no rust of the metal oxides or the metal that is the irons that we find in early rocks. And volcanologists have gone up and collected the gases coming out of volcanoes, and there's a lot of ammonia, there's sulfur, there's phosphorus, and there's all sorts of things coming out of those volcanic gases. This experiment has been repeated over and over and over and over again with different combinations of gases, with different types of heating sources, all sorts of different things, without lightning. And always, as they run this, there is chemical evolution occurring in these systems where we go from inorganic molecules such as water and nitrogen, or excuse me, ammonia and hydrogen gas and things like that, into organic compounds such as amino acids. And as I say up here on this slide, in 2007, scientists re-examined some of Stanley Miller's sealed samples. So these things were over 60 years old and hadn't been analyzed because Stanley Miller didn't quite have the analytical tools that we have today. And they found more than 20 different amino acids in these samples. What are amino acids? They are the building blocks of proteins. And that's a really important organic molecule, of course, that makes life possible. And so this was kind of a look into the possible formation of life on this planet from what we call abiogenic origins, i.e., origins or genesis without life, abiogenesis. Okay. There are some other theories out there. One's called panspermia, where scientists have hypothesized that life may have been seeded from outer space and on asteroids and meteors and things of that nature. And this is not very far-fetched because we have found amino acids on meteorites and asteroids. All right, so all organic compounds, like I said before, does contain carbon plus one or more additional elements, and one of those has to be hydrogen, okay, that are covalently bonded to each other. Now, there are many compounds, molecules out there that do not have, that are just that contain carbon, but they are not considered organic, such as carbon dioxide. Okay, or carbon like graphite, which is this right here is a piece of graphite down in the lower corner, and here is diamonds. These are both pure forms of carbon. Okay, and remember, carbon has an atomic number of 6. That means 2 electrons on the inner shell and 4 valence electrons. Those 4 valence electrons allow carbon to form 4 covalent bonds. Okay, just to show you real quickly, both graphite and diamond are both composed of pure carbon, and that's it. It's just that the carbon molecules, are bonded together differently. In graphite, the carbon molecules are bonded together in planes, horizontal planes. And so when you write with a pencil on a piece of paper, these planes of carbon are shearing off and being left on your paper. Diamond, the hardest substance known to man, the carbon molecules are intricately bounded in a 3-dimensional pattern, so they're all bound to each other and making it very, very stable and very, very resistant to any kind of impacts. Remember, tetravalence. So carbon can form 1, 2, 3, 4 covalent bonds. These are different models. This is a structural formula model. This is a ball and stick model, and this is a space filling model. This is what we were using the other day when we were doing the looking at water molecules. Here's this is ethane and this is ethylene, and the difference being that there's a double bond here, and we're missing 2 hydrogen molecules. Okay, so the formula for this is C2H4, where for ethane it's C2H6, and for the simplest organic molecule, methane, CH4. All right, so as I said the other day when I was talking about basic chemistry, the action happens in the valence electrons. In large molecules, the action happens on what we call functional groups. These are little combinations of elements that are a part of a much larger molecular structure. Okay, and we'll look at these in a little bit. But we're going to talk about all these different groups. There's a diagram in your textbook on page, I think it's 53 and 54 or 52 and 53, somewhere in there, that basically describes what all of these functional groups are. You should look at that. But these functional groups are really, really important. Here we see this large molecular structure. This is a steroid. This is basically estrogen, okay? And it's a steroid hormone. This is testosterone. It's another steroid hormone. You can see that the large molecular structure is basically the same shape, as are most of the steroid hormones. They are all derived from cholesterol, which you might go, oh, that's bad stuff. Cholesterol is extremely important for us in our cell membranes to keep them fluid at cold temperatures and to keep them together at warm temperatures. And it also acts as the precursor to all of these important hormones that we have. But let's look at these functional groups. Here is a hydroxyl group, and here is a methyl group. Well, the addition of the methyl group and the subtraction of a hydrogen turns estradiol into testosterone. This is the male sex hormone. This is the female sex hormone. They look pretty much the same, but boy, they sure have some differences now, don't they? Okay, so functional groups, really, really important. Carboxyl group is one of the ones we're going to be seeing a lot of because it's part of an amino acid. Okay, and functional groups do give properties to the large molecules they are. The carboxyl group, which is often written as COOH, okay, acts as an acid. Why? Because what defines an acid? An acid is defined as anything that can increase the proton concentration of a solution. So let's look at carboxyl down here in this formula. This is the non-ionized form. It's COOH. But when you put it in water, oftentimes this hydrogen is going to go to the water. So what has that done? That has now increased the proton concentration of the solution, i.e. it is an acid. It's a weak acid, but it is an acid. And so we call these carboxylic acids. Okay, and when we get talking about fats and things like that, and amino acids, we'll see that there's carboxyl groups on pretty much all the fatty acids, and all the amino acids have them too. All right. The amino group, okay, amino groups act as a base. It's NH2. That is the amino group. Why do they act as a base? What is a base? A base is something that can reduce the proton concentration of a solution. And when you put an amino group into water, or a solution that has excess protons, it takes that proton right here. It becomes ionized, and it also gains a positive charge. It's reduced the proton concentration of the water, so it acts as a base. And as you can see, here is an amino acid. We have a carboxyl group on one end and an amine group on the other, amino acid. So the name makes sense. Sulfhydryl groups, we'll talk about these more when we talk about proteins, but they're similar to an OH group. They can make a molecule polar in water, and they play a big role in the structure of proteins, because they can actually form a covalent bond from one amino acid to another in something that we call a disulfide bridge. Di meaning two, sulfur meaning sulfide, meaning the sulfur molecules there. So that's where they are important. Phosphate groups, we'll talk a lot about phosphates. Main function of phosphate groups is in the transfer of energy. Think of adenosine triphosphate, ATP. Okay, so here is that phosphate group right here. This is the way it is written. You can see it has a bunch of negative charges on there. Phosphates always have negative charges, and DNA has the sides of the DNA, what we call the sides of the ladder, of the DNA ladder, are made up of sugar phosphate groups. And because of that, DNA overall has a very negative charge because of those negative phosphate groups. All right, so the other thing is that all macromolecules are polymers. Macro, big molecule. Poly means many. Mer means part. So a dimer is two parts. A polymer is many parts. And a monomer is one part. Okay, so again, we can build a whole variety of polymers from a small set of monomers. So we can make an unlimited type group, unlimited number of different types of proteins from the 20 amino acid monomers that are found in every single living organism. And that's a big piece of evidence for the unity and diversity of life. Everything from a bacteria to a blue whale to you and I all utilize the same subset of 20 amino acids. There are literally hundreds, maybe even thousands of different types of amino acids, but all life uses the same subset of 20. And there are, I should say, there's a few exceptions to that in the archaea, but really, that's such a small number, okay? And I want you to point out up here, classes of biological macromolecules differ in the nature of their monomers, but chemical mechanisms used to make and break polymers are essentially the same. And here's the two of them. Hopefully some of you guys remember this from 9th grade biology. We have dehydration synthesis, which builds molecules, and hydrolysis, which breaks molecules. So let's look at these a little more deeply. Both of these involve water. Dehydration synthesis. So dehydration, when you're dehydrated, you've lost water. Synthesis means to put together. So we put together by taking water out. And hydrolysis, hydro means water, lysis means to split. So we use water to break apart. We can see the meaning in the word. And what we see up here, we see in a dehydration synthesis, we're going to take a water out. We're taking a hydrogen off of here and an OH group off of there. We're adding some energy because this is an energy-intensive reaction, and we make a bigger molecule. Now we want to break the molecule. We put water back in. There's my H, there's my second H, and there's my oxygen, H2O. We release energy in this process. So dehydration synthesis is an anabolic reaction. It's one where it's a building reaction. Think of bodybuilders, they take anabolic steroids to get bigger. Okay, you wouldn't want to take catabolic steroids, and you get really, really, really small. So anabolic is building. Endergonic means you're putting energy in. Hydrolysis is also a catabolic reaction. Catabolic or catabolism means to break apart, and exergonic that means energy is leaving. Like think of exit, you're leaving, going out someplace. Exergonic energy is leaving the system. Okay, or I should say, it's leaving the molecule. All right. So, like I said, once again, an immense variety of polymers can be built from a small set of monomers. And again, every cell has literally thousands, tens of thousands of different types of molecules in it, all built from a small subset of 40 to 50 common monomers. Okay, just think about, like, my analogy is you got 26 letters in the alphabet, and you can make as many different words as you want. Okay, and so biological macromolecules, which we'll get into next week pretty heavily, 4 categories of biological molecules: carbohydrates, which are basically short-term energy storage, things like, think of sucrose, glucose, dextrose. And also as structural materials. Think of cellulose, which makes up wood. Wood is made up of a whole bunch of glucose molecules. That's all it really is. Lipids, which are your fats and waxes and oils. They're kind of diverse in structure, but one thing they all have in common is that they are hydrophobic or water fearing, i.e., they are non-polar in nature. Water's polar, lipids are non-polar. Proteins, okay, these are the workhorses of your body. They do all the stuff. They make up your muscles. They make up your connective tissues. They're enzymes. They do just about everything. Your hair, your fingernails, all of that is proteins. Most of your skin and things. And nucleic acids, which are the molecules of information transfer. You're familiar with DNA, and I'm sure you're familiar with RNA in terms of messenger RNA and transfer RNA and ribosomal RNA, but there's a whole bunch of other RNAs out there, small interfering RNAs. Now I'm forgetting them all, but there's a lot of them out there, and we'll get into those in a lot more detail in the future. And that's it, guys. Hope this helps.